Hello again, biology class. This is Mr. B, and we are getting ready to start our second flipped lesson. And this is going to be on Chapter 1, Section 2, and we're going to look at how scientists work. So, last time we figured out what science was, we got a definition of that, got some steps to follow to kind of get us going on what science actually entails. So, today we're going to actually look at what the scientists do. So, we concluded our video talking about a hypothesis and maybe how to test those. So, whenever possible, a hypothesis should be tested by an experiment in which only one variable is changed at a time. All other variables should be kept unchanged or controlled. So a variable are the things that we're going to test for. So when we break it down, there are two types of variables. There's one that is deliberately changed, and that's called the manipulated variable. So for example, if we wanted to look at the way plants grow, and we wanted to see how sunlight affected them. So we would have two plants and we would give them all the same treatment except for the amount of sunlight they receive. So that would be our manipulated variable, the thing that's different between the two of them. So again, if we had a plant, we have two, they're both getting the same amount of water, they're in the same type of soil, they're in the same type of pot, they're in the same type of temperature, they're just exposed to different amounts of sunlight each day. And so as a result of manipulating that variable, there's going to be a variable that is observed and that changes in response to the manipulated variable, and that's called the responding variable. So when we're going to look at plants, we're going to obviously want to see how tall they're going to grow, or maybe how many leaves they produce, or something like that. So again, just think of it as manipulated is what we're changing, and then responding is what we would really be measuring, what we would use our quantitative data to measure. So those are the parts of an experiment. So when designing an experiment, the process kind of has 5 steps. And its first step would be asking a question, which that kind of happens way before this process even way before this process even starts. There would really be no reason for us to kind of do any of these steps unless we had a question to ask. So after asking the question, we're going to form a hypothesis, then we're going to set up a controlled experiment, we're going to record and analyze our results, and then we're going to draw a conclusion. So to go through this designing an experiment, we're going to look at a very historical example, a very famous example of kind of biology at its finest very early on. So way back many years ago, people wanted to know how living things came into existence. So they asked, how do organisms come into being? So there's step one. They wanted to know where life came from. So they asked that question. So someone formed a hypothesis, and one early hypothesis was called spontaneous generation, and this was the idea that life could come from non-living matter. So basically life just appeared out of thin air, like came out of nowhere, almost like a genie said, let there be life, and there it was. So for example, a lot of people thought that maggots spontaneously appeared on meat. So you see that next year there's 1668. You have to think there's no refrigerators back then. So if meat gets left out, maggots are going to appear. So we know now today that that's not true, but back then they really didn't have the technology or the means that we have today. So they just kind of went with their gut, what they saw, and they're like, oh, well, maggots came out of that meat, so the meat must have gave rise to the maggots. However, in 1668, after making some observations, a guy by the last name of Redi, R-E-D-I, proposed a different hypothesis, and his was that maggots came from eggs that flies laid on meat. So this might, again, seem really simple to us, but back then that was a pretty radical statement to make something like that. So he set up an experiment, and there's a picture of it, what it would kind of look like. So it kind of breaks it down here. It starts with his observations that he made. So he saw that flies landed on meat that is uncovered, and later maggots appeared on that meat. So he made the hypothesis that flies produce maggots. So again, he didn't just come up with that on his own. He made a distinct observation. So he set up his experiment here with his controlled variables. The ones that aren't being changed are the jars. As you can see, they're both the same in both pictures there. The type of meat, the location, and the temperature and the time. Everything is the same except for what is being uncovered there. So uncovered meat, covered meat. That's the only difference. So as you can see, the flies are getting into the uncovered meat, and the flies are not able to get to the covered jars. So his data and conclusion, you can see, again, I just mentioned his manipulated variable was the gauze that was keeping the flies away from the meat. And then there's his responding variable where maggots appeared. So whether or not maggots appeared, and after several days, maggots were all over the meat that he left out, and there were maggots not anywhere near the ones that he had covered up. So he was able to record that data, and he shared it with the world saying, hey, listen everybody, I know this might sound crazy, but life doesn't just randomly appear. It has to be given, it has to come from another life source, because I just did this experiment with uncovering and covering meat, and maggots only appeared on the meat where flies landed. So he made the conclusion that maggots form only when flies come into contact with meat, and that spontaneous generation of maggots did not occur. So this was a big breakthrough. He followed some really definite steps here, and he came up with this conclusion that, again, today it's accepted, but back then people probably were pretty hesitant to accept this. But as we're going to see in tomorrow's video, this is a longer section, so we're going to break it up in halves. Scientists need to repeat experiments to be sure that the results match those already obtained. So we're going to see tomorrow some people that are going to challenge him and some people that are going to try to support him. And today, this is one of the biggest deals in science, is actually just repeating what's been already done, because we don't want to be stuck in the past. So if you have any questions, let me know, write them down, and we will get back at it tomorrow.